together with at BCIC, our mission is to build the bridge between US and China in a variety of areas, including biotech, finance, and investment. My name is Lin An. I'm part of the BCIC Biotech Department. Now I'm going to hand over to Dr. Michael Zhao, who's representing Cubase and will moderate the first session. Michael, all yours.
Thanks, Lin. Good night, everybody. Welcome to tonight's conference. I'm from Cubase Center, Boston. I'm the Cubase Center Boston partner. So let me give just a brief introduction about our company. Cubase is an organization that focuses on a global perspective of healthcare industry. We basically provide resources, including fundraising, connections, entrepreneurship, and innovations. Also, we provide resources, including biomedical incubators. We have a huge incubator in San Jose. We also have expert mentors, resources to provide, and also, of course, the financial support as well. So Cubase is consistently promoting collaborations between investors and startups. Simply speaking, we help innovations. So if you have any question or need any help on entrepreneurship, please contact us. Last but not least, I wanted to thank all of my Cubase colleagues to make this happen as well. Dan, Brian, Yanlu, Yichen, and Tianyi. Without your help, none of this would happen. Okay, let's jump into the show tonight. Today we have 7 pitching companies in total. I'll be the moderator for the first 4, and my colleague Lin An is going to take it over. So the first company we have tonight is Iclos. The Iclos is a biopharmaceutical company. On a clinical, it's a clinical company focusing on translation and innovative science into treatment. The company currently is developing novel non-narcotic drugs for treatment of neuropathic pain. Today, the founder and CEO, Stephen Fox, is going to give us an introduction about it. Stephen, if you're here, I can share the slide for you.
Great.
Hang on a minute. And yeah, you have to stop screen. Yeah. Are you able to see that?
Yes, I can. So Iclos is a first-in-class HCN1 ion channel inhibitor for the management of neuropathic pain. I'm hearing somebody else speaking. Oh, are we okay? Can you hear me all right? Next slide. Hello? Yes, you can skip past this slide. So neuropathic pain is caused by a few different kinds of ailments. It could be diabetic pain, CIPN, chemotherapy, multiple sclerosis, stroke, post-surgical pain, atypical facial pain, shingles. It's very debilitating. And it's very different than acute pain. It's something that you wake up with every day, and it makes life very, very difficult. Next slide. Our first targeted indication in our clinical studies will be for CIPN. CIPN is very, very difficult on a patient. If they're taking chemotherapy and you open a window, even the breeze could make them just in incredible pain, and they would rather almost end their chemotherapy and die than live with the kind of pain they get from chemotherapy. So it's very, very common for people to have tremendous pain with CIPN. Next slide. So it's a global problem. Cancer is with us. There are almost 2 million new cancer cases every year. It will rise 70% over the next 2 decades. And the CIPN affects 90% of the patients. And there's no real meaningful drug to help a patient with neuropathic pain. Physicians and dentists have no prescription that they can write that will work. And unfortunately, we have them writing opioids, and we are now in the worst pandemic, not COVID, but the opioid crisis. That's the worst pandemic in the history of the United States, and accelerating every day. Next slide. The market opportunity is huge. You'll see here that it's $8 billion by 2023, but I just got a new prediction, and it's up to $25 billion per year. So the market opportunity is unlike anything we can imagine. Next slide. So as I said before, there are very few prescriptions that work. In fact, none work for neuropathic pain. Physicians and dentists use anticonvulsants, antidepressants, and opioids, and they simply don't work. Next slide, they're placebos. You can see that an example of that is fluoxetine or gabapentin. They have tremendous revenues, billions and billions of dollars, and in combination with opioids, even more billions of dollars, but they simply don't work. Next slide. So the problem, as I said, is that neuropathic pain is with us. It's not going away. It affects up to 500 million people worldwide. Next slide. So we've been able to target the HCN1 ion channel, and we know that it's expressed in rat models from from injuries in the rat models. Next slide. And paclitaxel, which is used in chemotherapy, significantly increases the activity of the HCN1 ion channel. Next slide. So if a rat in a study is exposed to cold or any kind of thermal exposure, we can see the HCN1 ion channel again increase in terms of its activity. Next slide. So in humans, the HCN1 mRNA is present in a larger proportion than HCN2. So it's all over the peripheral nervous system, and we think that's the best target. I also might add that HCN2 probably has cardiac implications, and we don't think it'll be as good a target or as safe a target. Next slide. We are, our strategy is a platform anchor tether pharmacophore model where we anchor the pharmacophore exactly where it needs to be at the HCN1 ion channel in the cell membrane. We're also working on another strategy to couple that anchor with monoclonal antibodies. Next slide. And what we know from working in our laboratories is that HCN1 is expressed in a concentration-dependent manner. It's inhibited in a concentration-dependent manner. Next. Our triple-blinded studies where we've worked on nerve injury models with rats have shown that with mechanical stimulation or thermal stimulation, that as you increase the dose of our drug, you reduce the amount of sensitivity, you reduce the amount of chronic pain, neuropathic pain. Next slide. Next. And these are standard models, as I say. And we've also shown, you can go to the next one. We've also shown that we have tremendous bioavailability in our triple-blinded studies that were done at Weill Cornell recently. This technology has been licensed from Weill Cornell and Columbia University. And they put $9 million into it over the last decade. In addition to that, we have an NIH grant. There were only 5 companies in the United States under the HEAL Act that received substantive grants from the federal government of the United States. Next slide. So it's effective, it's bioavailable, it's potent, and it's an effective inhibitor of HCN1. It anchors the lipid membrane, and it relieves mechanical and thermal hypersensitivity in male and female rats. It's extremely safe. It doses 10 times the lowest efficacious dose. It doesn't produce any changes in heart rate or blood pressure. It does not produce changes in motor strength, coordination, or activity. It's non-sedating and does not show potential for abuse, addiction, which is a hallmark of opioids. So we are a non-addictive, non-opioid drug because we don't cross the blood-brain barrier because we have this anchor tether pharmacophore model. Next slide. We have an exclusive license with Weill Cornell University. We've shown it to be safe and effective. We have our grants, as I alluded to before. We're looking for a $14 million investment. And we think that'll take us to phase 1 testing. Next slide. I think we have the finest scientific and leadership team in the United States. Mike Levitt won the Nobel Prize in Chemistry. Chuck Birdie is the Chief of Pain at Harvard Medical School. We have 4 researchers at Weill Cornell Medicine, Dr. Goldstein, Dr. Warren, Dr. Willis, and Dr. Tibbs, who are leading the research. Next slide. In addition, Roger Frechette has developed drugs over the last 30 years. I was on the faculty at Harvard and NYU. I'm a doctor of dental surgery. I was Ernst & Young Entrepreneur of the Year, received the Medal of Freedom from the United States Senate. And I was chairman of a company that in 1997 became the best performing stock in the United States. And we changed oral health care all over the world. Peter Hutt was a former chief counsel to the FDA. Roger Frechette, as I alluded to before, has developed drugs over 30 years. Bob Benson was head of licensing and technology at Harvard University. Senator Thomas Daschle was the Senate Majority Leader of the United States, a healthcare icon. And Tom Hefner developed Lyrica as head of neuroscience for Pfizer. Thank you for your time. Sheshe. Any questions?
Is there any questions from the audience? You can raise up your hand, and then I can pull up you to the speaker. Otherwise, you wouldn't be able to speak. Any questions from the audience? Please raise up your hand. Okay. Or and also you can ask the question through the chat box. Send the message directly to the chat box, and I can read that for you if you don't want it to speak directly.
Or you could email me anytime or call me anytime.
Sure.
That's fine too.
Is there any questions? I have a question. So if I'm understanding correctly, the AKE 1018 is probably inhibit HCN1, right?
Correct.
So, but is there any like biochemical data like SPR, DSF, I mean, to show how AKE 1018 is inhibiting HCN1 and efficiency? So any biomedical data to show that? Thank you.
Well, I mean, the medical data that we have shows that we're getting the results that we're looking for. Any specific details on that, I really would leave for Dr. Goldstein to answer the questions, and I can arrange that because he's the lead scientist and he knows how to answer those in greater detail. I couldn't give you more detail than I have tonight.
Yeah, but are you planning to do that, or it's
Well, we're planning to do everything. Toxicity studies, pharmacokinetic studies. We're doing all of the tests that would be required of us to get to an IND, and then to go through phase 1, 2, and 3 in order to get FDA approval. So we'll do every test that's required for a new drug with a contract research organization.
Okay, sure. Thanks. There's a question from the audience. Li Bin Cui, Biotech, is asking if there any IO related pain, same as DCIP. IO related pain, I think it's intraosseous. Is that right? If I'm understanding correctly. Yeah, please use the full spelling for the word. So the question is there any IO related pain? Same as DCIP.
IO related pain. I'm not sure what you mean by what
yeah.
IO stands
I'm
for.
also guessing. I mean, Li Bin, if you're there, would you mind to like make it more specific? I'm understanding as intraosseous.
Well, look, let me just answer it this way.
Is that intraosseous? Thanks. Oh, you raised your hand. Hang on a minute. Let me allow him. So, yeah, you should be able to talk.
Okay. Yeah, thanks. So yes, so I'm asking the question because I have a friend who has cancer and who was treated with PD-1, and after the treatment, he has lots of pain. I think is neuro-related pain. So I want to
Right.
know whether it is similar as the chemical induced neuropathy. Thanks.
I didn't catch the end of the question. You want to know what?
I want to know whether the mechanism is the same. The immunotherapy caused pain is the same as the chemical induced pain.
The way that the chronic pain will be treated will be for somebody to take a pill, hopefully once or twice a day. It might even be in injection form. We're going to be targeting CIPN initially, diabetic pain, shingles, post-surgical pain, atypical facial pain, anybody that has neuropathic chronic pain. So I hope that answers your question. I don't.
Yeah, I think you're targeting the target that is related to chemotherapy induced neuropathy, and this caused the pain of the patient, right? So yeah, so I want to know whether there's a similar mechanism of the immunotherapy caused the pain neuropathy.
The mechanism is to inhibit the HCM1 ion channel.
Okay.
That's the mechanism of action.
So is there a related, is there any report showing that the immunotherapy also affects the expression of this channel?
I still don't understand. I'm sorry, your English.
Let me translate. I think I do understand a little bit. I think Levin is trying to ask it for like the oncology pain, since like the chemo pain. Is it
Right.
similar, or it's can you do something about that? So the question is like that.
Well, that's exactly what we're trying to do. CIPN is chemotherapy-induced peripheral neuralgia. So what we're trying to do is inhibit that. So anything induced by chemotherapy, we're trying to inhibit that pain.
Okay. There's another question from the chat box. Wade Lee is asking, is there any addiction potential? I mean, it's actually an interesting question.
No,
and also any data available from animal model mimic to chemo induced pain?
Well, the whole point of this is that it's not going to cross the blood-brain barrier. And that's how you don't have addiction. It'll be a non-opioid, non-addictive drug. This will be the first non-opioid, non-addictive drug for neuropathic pain. So therefore, there'll be no addiction. And we also think that we'll solve the opioid crisis.
Thank you. Thank you. Any other questions?
I have a question.
Yeah, go ahead.
Yeah, can you give some comment about your antibody candidate related to the drug candidate? Thanks.
Can we tell? I'm sorry. Are you asking if I can disclose?
Yeah, can you little bit about
the
antibody candidate for this drug?
I can't disclose that. That's being worked on in the lab at Weill Cornell. It's not something I could disclose.
Thank you.
And Li Wei is asking a following question about the long-term toxic. Is there any long-term tox?
Well, we're going to be working on that. That's part of those studies, toxicology studies. So far, no. But we have other toxicology studies we'll be doing as we approach an IND. So we'll be going through all of that. But so far, it's been extremely safe.
Okay, thank you. I think the time is almost up. So really appreciate Stephen. So if you guys have any questions, please send email to me or to Stephen directly.
Sure.
Get connected. So I'm sure there have tons of questions. So please contact us later. I really appreciate Stephen.
My pleasure. Shay.
Really nice presentation. Thank you. Have a good night.
Thank you. Take care.
Okay, I'm gonna stop this first. The second company we have tonight is Matrix Therapeutics. So Matrix Therapeutics is a biopharmaceutical company that advances the next generation of new anti-inflammatory peptide therapies for patients with chronic inflammatory diseases. The company's product AMTX100 is being bioengineered using human protein sequences that modulate the immune system by natural process. Tonight, the CBO, Randall Briggs, is going to talk about it. Randy, you here?
Yes, can you hear me?
Yeah, please go
Okay,
ahead.
great.
Floor is yours. Thank you.
Can you see the slides?
We can see the slides, but your
Okay.
mic is probably it's a little bit blurry. I mean, it's you want to adjust your mic a little bit?
Okay, is that any better?
Yeah, it's a little bit better. Please go full screen.
Okay, I'm gonna try to, but I think it's in Mike, I think it's in PDF version. Let me see if I can get it full screen.
It's the slides. You can go full screen.
Okay,
The bottom,
put it
the
in full screen.
bottom right.
The bottom right,
A little bit left.
left, okay.
Or you can go to the ribbon slideshow. Slideshow. Yeah.
Okay.
There you go.
Trying to get this off. I don't know what that is doing there, but okay. So, oh, well. Now it just did something else.
Yeah.
Technology, love it. Okay.
Go ahead.
So as he said, we're using the natural cellular process to develop an anti-inflammatory drug that is the first novel total human sequence chimeric peptide that has certain signal sequences that are conserved on the FGF side of the left side of the peptide, and then on the right side is a conserved region of the nuclear localized sequence on the other side of that sequence for the peptide, and that's NF-kappa beta. It's not the whole sequence, it's just the conserved sequences, and you'll see why we did what we did as I talk about the presentation. But I will say that innate immunity, I mean, we're talking about inflammation as us aging as society is becoming a huge issue, and it's causing a lot of illnesses from autoimmune diseases, arthritis, lupus, et cetera, and the market, no matter what is out there, whether you're looking at TNF antagonists that have black box warnings by the FDA saying that they can cause immunosuppression, cancer, make you more susceptible. These are big issues for these patients that are getting older. And I should say also in China is even an older aging society than America, which we are aging as well. So it's just becoming a huge need. That chronic uncontrolled inflammation is causing a lot of chronic diseases, and our product addresses that. And I should say too that we've already raised 18 million. This product I'm about to show you has already completed a phase 1, phase 2A program in humans. So it has already been FDA approved to go into humans. We finished the phase 1, phase 2, and it was very successful. There was no AEs or SAEs, none, zero. And 95% of the patients had a major biological response. And what we did is we did an atopic dermatitis because we wanted to see the results very quickly. So the drug works and it's very safe. And this is the picture of kind of the cartoon of the drug. And as you can see on the left side, you got certain human sequences of the that are conserved in all human sequences, amino acids of the human FGF4 domain. And then on the right side, the NF-kappa beta, certain conserved sequences, and a nuclear localized sequence. And what happens is, I want to explain this. We are not inhibiting like a small molecule or an antibody that binds to the target and stays on the target. We are not a covalent binder. What we do basically, and this is very important you understand this, is we only act as a decoy. because whenever you get pro-inflammatory transcription factors that get into the to the cytoplasm, they need in the cell, they need the important alpha beta complex to hitch up to them, to bind to them, and take them into the nuclear pore, and then they get dropped off into the nucleus, and then they basically activate cytokines and chemokines and growth factors. And all we do, all this drug does with that character, that cartoon I show you, is it makes the transporter of important alpha and beta think that it's taken a pro-inflammatory transcription factor into the nucleus to activate cytokines and chemokines and growth factors. And in fact, when it's taking our drug, it's a placebo, basically. It gets dropped off into the nucleus, and basically nothing happens. It gets chewed up and eaten as cellular metabolites and things of that nature, or energy, and it gets excreted out. So there's no damage done or interference with the NF-kappa beta or the human growth hormone, FGF4. Again, it's just a decoy. And what that does is it dilutes or modulates the pro-inflammatory transcription factors, basically to where it calms down the inflammation. This does not affect housekeeping genes, because housekeeping genes do not need a transporter. They go in and out of the cytoplasm to the nucleus and back out again. That's a very important distinction of our drug. And what this is showing here on the left side versus the right side, you just see basically the AMTX on the right side as the decoy. And on the left side, if it weren't there, you see the normal cellular process hooking up with the stress relief transcription factor and going into the important beta or alpha beta complex and being deposited into the nucleus, and then creating, again, these are pro-inflammatory transcription factors, creating chemokines, cytokines, and growth factors. And all we do as a decoy is we tell that basically transporter, hey, I look like a pro-inflammatory transcription factor, take me into the nucleus, and basically nothing happens, but it takes up time and therefore modulates and calms down the cell to where you get resolution. And that's really important. And there are a lot of different large transcription factors that basically, as a decoy, we basically reduce them going into the nucleus to create all those chemokines and cytokines that are bad. And I'm talking about chronic inflammation. And if you look at what we've done, again, I told you this is a phase 1, phase 2 completed drug, but I want to show you that before it got into humans, we did a ton of animal studies. And whether you're looking at acute lung disease models in animals, liver, diabetes 1, these are all inflammatory diseases, atherosclerosis, hypercholesteremia, skin, sepsis, even sepsis, which is a very difficult thing. And we actually, the mice lived with our drug versus the controls, they all died very quickly. The sepsis happens very quickly. But you can see, you can look up these things yourself if you want to. They're there, they're published. The drug works in animals, and it was very safe. And we've seen that in phase 1, phase 2 as well. This is the first, we did the first phase 1, phase 2 trial with atopic dermatitis, because again, we wanted to see the proof of concept in humans that it was safe and had biological activity based on the mechanism of action. This drug is formulated into a water-based, 87% water-based formulation. It looks like whipped cream. It's non-odor, it's not odor, it has no odor, it's non-sticky, and it's non-irritating. And you put it on, and within 2 minutes max, you don't even know you've put something on your skin, because again, it's 87% water-based, 11% active drug, and then 2% excipients. And the patients really loved it. We had no problem, even during COVID, enrolling patients because the doctor saw such phenomenal results. And we did not use steroids in our trial. That's very important. All the Dupixents and all those others, they always use steroids in addition to their treatment. We used only our drug. That's it, no steroids. So let me go to the next one. This is the phase 2. Sorry, I don't know what happened there. Let me go back here. This is the phase 2. How do I get this again to go to active screen? This is the phase 2 that you can see as I'm trying to find out how to get it there. There it is. Okay. This is the phase 2 results, phase 1, phase 2. And what you see on this, I'm not going to explain everything on this, but we had 5 cohorts, and the different colorations are the different cohorts at the same dose, but covering the body skin with the atopic dermatitis drug up to 10%, all the way up to 60% of the body covered in cream. And what you see on this right side with moderate to severe, or I'm sorry, mild to moderate, we had, even in those very difficult patients that had 60% of their body covered with atopic dermatitis, they had, like in the number 1, almost clear with just our drug. And I should point out, this is only treatment of 1 week, 2 times a day. And then after 1 week, we stopped. And then the patient was evaluated by the physician at the stop of the treatment. And you can see the results are extremely positive. And then 2 weeks later, they came in after treatment, no more treatment, and they still had resolution and almost clear skin. And we think that's kind of resetting the immune system based on our novel mechanism of action. That's the only way you can explain it, because they didn't get drugs. And most of the time, Dupixent, all of them use it for 16 weeks and with steroids, and they don't have as good as results as we have with just 1 drug for 7 days, applied twice a day. So basically, I just want to point this last slide out. We've already, like I said, raised 18 million, and we've done extremely well. With that money, we still have money left over. We're looking to do things in China and to Japan and America and Europe to do basically not only studies in atopic dermatitis, but in lupus, because you can easily formulate this drug to an oral, and we have also a subcutaneous delivery as well as IV delivery. And also, I just started working with a company that has a formulation for our drug that is called thin film freeze drug, that you can inhale the drug with their own device. And it's nanoparticles, and they've already formulated it, and the bioactivity is still active. And so we can treat COPD, COVID, and things of that nature that results from cytokine release syndrome or overinflammation. And that's kind of where we're at with this.
Hi, Randy. You muted yourself. You want to unmute it?
Okay, we basically think that, again, the patent situation is a long time till 2037 competition, and we believe that with lupus and all these other indications for anything with inflammation, we believe with our different formulations, whether it be IV, oral, subcutaneous, we're going to be able to really address a huge unmet medical need in inflammation and not have immunosuppression and toxicity like you see with today's current medicines. So I thank you for your time, and that concludes my presentation.
Thank you, Randy. It's a really nice talk. Is there any questions from the audience? You can always raise up your hand. I can allow you to speak, and also alternatively, you can type your questions in the chat box. We can read it for you. Is there any questions? I can start with one. So you just talked, I mean, probably already covered that. I see that the administration is IP and IV, right? Is there any stability study being done, like the PKPD study in the animals especially? Thank you.
Yes, thank you for the question. We have over 3 years of stability of the drug.
What's the half-life?
Oh, great question. We just did biodistribution studies, which we know the drug goes into immune cells specifically, so it's precision-based medicine, which is why we're getting good results. The half-life is anywhere from 45 to 51 hours. Have you ever heard of a peptide that has that long of a half-life? It is amazing.
This is very, very long, couple hours. I mean, in general, if it's over 1 hour, is already good enough in general for any, even proteins.
Yep, and we're 45 to 51 hours. Amazing.
Yep. There's another question in the chat box. It's asking if the NF-kappa B, the only transcription factors you're tracking for.
No, absolutely not. Basically, again, you got to remember back at the mechanism. This is so novel, and I know people are focused on the nuclear localized sequence on the NF-kappa B, but it's not just those. NF-kappa B is not your only large transcription factor, pro-inflammatory. There's also STAT1, AP1, there's a ton of them. And again, you remember, all we're doing is basically as a decoy to the mechanism that grabs these pro-inflammatory transcription factors. It doesn't matter if they're NF-kappa beta, AP1, STAT1, it doesn't matter. We basically look like what that transporter needs to grab in order to take it to the nuclear pore and then deposit it into the nucleus to then activate chemokines, cytokines, and growth factors. We basically are the decoy that modulates that process. We don't stop it. So it's still a normal cellular process. We just preoccupy the transporter so that it thinks it's taken in the big transcription factor when in fact, it's taken in a placebo. And that in of itself makes the modulation of inflammation go down. And we've seen it in animals, and now we're seeing it in our phase 1, phase 2 studies phenomenally with just one product, no steroids, just by itself, and only for 7 days of treatment.
Yeah, I just wanted to ask a follow-up question with that, the NF-kappa B. So because we all know NF-kappa B is very long, so are there any biomarkers or double confirmed that is really inhibit NF-kappa B? So any like P64, I mean P50, P65, or IKK, IKkappa B, any biomarkers or double confirmed that is really inhibiting and the efficiency is high? Thank you.
So good question. Again, I think I would just, again, this is such a novel mechanism. I think people have a hard time understanding that we don't need to have a biomarker. We just need to make sure that our decoy is attractive to the transporter. That is a critical process to take large pro-inflammatory transcription factors into the nucleus to activate chemokines and cytokines. And we see that through the physiology of what we've done in animals and in humans and in different disease states. So one thing I can tell you for our phase 2B, as in boy, that we're doing, we are going to use a company called it's DermTech. They do a smart strip, and they take a piece of I guess it's like tape, and they put it on the patient's skin and they rip it off, and then they basically do that 3 times, and they get the RNA and stuff, and they're able to find out which DNA genes, which genes are upregulated, downregulated. And then we're going to basically treat those patients, and we'll be able to see which genes go down and which genes go up, and then which patients have physiological good responses to our drug. And that way, when we go to pharmaceutical companies, we'll be able to say, these patients with this genotype will respond extremely well to our drug. And so we are doing, again, this is all precision-based medicine. I will say again, remember, this drug gets into immune cells specifically, which is a great thing. And I even know which specific immune cells it goes into, but I can't tell you that because we're filing a patent on that. That's confidential information.
Thank you. There are 2 questions in the chat box. I think one that's asking, as a peptide, will it be digested if administration by orally route? Another question is related, actually, is asking for toxicity. So any concerns addressed for the immune reaction for the synthetic peptide? Any immune responses? Thank you.
So I'll answer good questions. The immunosuppression or immunoresponse, it's all human sequences, and it's all conserved human sequences. So in the animals, it's amazing. There was no immune response even in animals, nor was there any SAEs or AEs in humans. And so what that tells us is these are very conserved sequences, and they're all made of human sequences. So no, we have not seen that. The digestive part of the stomach, we do have formulations that bypass that and will prevent that with the acid in the stomach. But again, we also have very good formulations for subcutaneous injection as well as IV, and also nose sprays of all things. I know you're saying, why are you running the nose spray? Well, turns out that Alzheimer's, Parkinson's, if you look at the key thought leaders now, they're all saying that inflammation from the microglia cells in the brain, and ours does pass the blood-brain barrier, is what's causing a lot of the actual upstage issues with Alzheimer's and Parkinson's. So we also can address, I think, in the next wave of how you really address Alzheimer's and Parkinson's, is to basically do what we're doing, which is basically address the early part of inflammation that leads to a lot of chaos in the brain and damage.
Thank you. I think there are still a lot of questions asking the BBB, asking the diet fluency, the efficacy, but due to the time, we probably just, if you have any question, please just send an email to me and I'll, or send to Randy directly so we can communicate later. Thanks a lot, guys. And thanks, Randy. It's a really nice talk. It's a wonderful presentation. Have a good night. Thank you.
Thank you, you too.
Yeah. Our third company tonight is going to be Neologian, Oncosystems. So Neologian is a contract research organization that offers the cancer drug development services for pharmaceutical and biotechnology companies. So Neologian performs corrective studies between drug responses and multiple tumor microenvironment to develop diagnostic that can be applied to clinical trials and patient care. Today, the VP of the Business Development and Marketing, John Waterman-Smith, is going to talk about it. John, you can take the floor over. Thank you.
Thanks, Michael, and thank you to Cube for the invitation to talk today. Much appreciated. Hopefully everyone can see my screen.
Yeah.
Okay, excellent. Thank you. So just to start, we're, as Michael mentioned, we're a CRO. We're based out of Tampa in Florida in the US. And we currently work with a wide variety of clients globally, from large pharma all the way through to small to mid-sized biotech. And really, I wanted to start by talking about where do we fit into the whole process. So in terms of where Neologian technology is used today, in the discovery side of the pharmaceutical business, we work with a wide variety of companies, screening therapeutics, and this is with real patient samples, patient tissue, fresh patient tissue, which I'll talk about in the oncology field, and everything from the small molecules all the way through to oncolytic viruses, everything's being tested in our system. In the development phase and translational studies, we're being used by our clients as a way to translate the molecules out into the clinic, looking at everything from mechanism of action all the way through to efficacy and dose escalation studies. And then in the co-clinical arena, essentially what we're doing is we're helping our clients who basically tested the technology in the development field to move out into their phase 1, phase 2 studies, looking at everything from patient selection, enriching their trials, and actually, as we'll talk about today, looking at providing the ability to help patients avoid things like futile therapeutic interventions and help guide treatment options. Sorry. So in terms of the issues today, we're going to focus on 2 areas from an oncology perspective in terms of the development dilemma and responder crisis. As we'll briefly touch on in a moment, the big issue really in oncology development is the fact that 95% of drugs still fail in clinical trials. And we certainly help address that with our clients in terms of testing their molecules before they go into the clinic. There's also a massive responder crisis, as kind of been alluded to already by a couple of speakers. So typically today, right across a variety of different oncology indications, 80% of the drugs that are used actually don't work. And in terms of the numbers here, you can look at the publications that occur. Really, things haven't dramatically moved forward in the last few years. We still have a very poor success rate of cancer drugs in the clinic. And of those, what you see is about 80% of those fail due to efficacy or safety issues, and about half of those is purely down to efficacy. And really, efficacy is one of the major reasons for late-stage drug development failure. So at Neologian, how can we help with this issue? Well, really, first of all, it's important to understand what the big issue is. So really, the tumor microenvironment is an incredibly complex 3-dimensional space. And when you're evaluating therapeutics, we need to take into consideration the variability of tumor cells, as well as the impact of the hundreds of proteins within the extracellular matrix, the infiltrating and resident leukocytes, and the stroma. So while intratumoral variability occurs between patients and is an important factor to be considered in clinical trials, intratumoral heterogeneity also needs to be addressed when you're evaluating therapies. And you can see on this particular slide, on the right-hand portion of the slide, we've got a H&E of a lung adenocarcinoma. And on the right side of that image, you have a zoomed-in area, which show many tumor cells, but few to no leukocytes. Whereas on the left-hand side of the tumor, it contains many tertiary lymphoid structures, and the zoomed-in area shows nearly all immune cells. And really, evaluating this tumor using things like a punch biopsy or a tissue slice, really depending on where the material was taken from, might classify the tumor as hot or cold. And therefore, using tissue slices to evaluate a therapy would not capture the heterogeneity of the complete tumor tissue. And in addition to the complexity and variability, we've also got to take into consideration the stromal environment. And the stromal environment within humans is very unique in the fact that it takes years to form through the continuous wound healing process. And eventually a pro-tumoral environment is created with the generation of cancer-associated fibroblasts and the secretion of chemokines and cytokines, which can result in the promotion of malignant growth, angiogenesis, and cancer metastasis. And the whole process may take several years to develop in the human. And so models which develop in days to weeks simply can't represent the impact that stroma, extracellular matrix, and the complex populations of immune and tumor cells have on the ability of a therapeutic to both penetrate the tumor and then activate the immune compartment and kill tumor cells. So this is where Neologian's 3D tumoroid platform provides a very unique approach for drug development. And at Neologian, we always begin with fresh, never frozen patient tumor tissue that's been resected. And within 24 hours of resection, the tumor tissue is generated into 3D tumoroids. The generation process itself takes all of 3 minutes and is purely mechanical in nature. There's no chemical dissociation, no cellular propagation, or organoid reassembly involved in this process. And for many tissue types, such as colorectal cancer, renal cell carcinoma, we typically receive about 300 milligrams of tissue, which when processed creates 9,000 tumoroids, all of the same size and shape and about 150 microns in diameter. And with these thousands of tumoroids, we can capture the heterogeneity of the tumor itself whilst preserving the intact tumor microenvironment, retaining all the structural components, the cell-cell and the cellular matrix interactions. So when we pool thousands of tumoroids to create a homogeneous population, we plate them for assays, and we use aliquots of about 400 tumoroids for the various assays that we use for things like flow and RNA, and about 100 tumoroids are used in the confocal microscope assays. So once plated, we perform QC via flow cytometry to ensure tumoroid viability and evaluate the frequency of immune cells. And at this point, we can also include markers in our flow cytometry panel if a client wishes to evaluate inclusion/exclusion criteria of a patient onto a study. So with tumoroids, we preserve the structural components of the original patient tumor within the 3D tumoroids. And on the left-hand side of the screen here, we've got a H&E of the original patient tumor and our adenocarcinoma of the lung. And on the right-hand side, we've got a H&E of the tumoroid that was generated from the tumor, where you can see the actual tumoroid itself and all the associated cancer-associated fibroblasts, tumor cells, and even tumor-infiltrating leukocytes. And because the generation of our tumoroids uses no chemical dissociation, there's no cellular propagation or organoid reassembly, we preserve the structural components and the cell-cell and cell extracellular matrix interactions of the original patient tumor. So Neologian has a clear certified labs based in Tampa in Florida, where we process clinical samples, but we also have a very large network of tissue providers in the US, at least one in every state in blue on the map. And all the providers are within the US, the tissues are ethically sourced and the patients are correctly consented. And we have a significant experience with all the tumoroids listed on the right-hand side of the column. And all the tumors we receive are treatment naive. We also source heme cancers, and these come with a tumor microenvironment, which is in most cases bone marrow. So 3D Explore, basically we use the tumoroids that we create, and we have a very holistic approach to the complete discovery process. For the sake of time today, I'm just going to touch on two of our unique assays that essentially use confocal microscopy, but we use flow cytometry. We do a lot of RNA sequencing or nanostring, and also single cell RNA-seq and proteogenomics. And we can always talk to people in more depth about this another time. In terms of confocal, just briefly to mention if people aren't familiar with this, essentially with confocal microscopy, what we're doing is looking at these 150 micron tumoroids, and we're taking essentially Z-planed 5-micron slices through this visually to capture all the fluorescent information that's in these images. And then we essentially put these together. We create a 2D stack, then generate a composite image and do the analysis of this data. So the two key things we're going to talk about briefly today are penetration and then tumor cell killing. So with penetration, what we're doing is we're looking at here. Sorry, I have someone, Michael's raised his hand. Sorry. We can look at everything from oncolytic viruses to CAR T cells, antibodies, and small molecules that have basically been fluorescently labeled. And we can track and trace these as they move into the tumor microenvironment. So a regular concern that is often talked to by our clients is, will my therapeutic be able to reach the target? How will I know? And in many cases, the dense stromal and extracellular matrix can prevent the penetration of the drug to its intended target. In example images, here we're looking at engineered T cells that have been labeled as cell tracker. And if we look at the images at the bottom here, we can see how, in this case, 2 of the tumors, we've seen very effective penetration of the T cells into these, whereas tumor 3, there's very little penetration, and we can capture this. So Michael keeps raising his hand, and I'm not sure I can see what that's about. I'll keep talking for the moment.
You go ahead and talking now, I can take care of it.
Okay, all right. Sorry about that, hold on. So with tumor cell killing, again, we use the confocal microscopy, but what we do is we visualize and quantify the tumor cell killing using high content confocal microscopy. In this particular example, we're looking at ovarian serous carcinoma, and it's been treated with a PARP inhibitor, and we're comparing a control to the treated arms of this study. So to detect live cells, essentially we add a proprietary viability stain at time 0, and then we're visualizing the tumoroids with a confocal microscope. We acquire the 5-micron Z-planes, and at the end of the study, we use our proprietary dead stain. And those cells that are dual labeled with both the live and the dead stain, we measure at the end of the study, and we quantify this number. So briefly, I just wanted to talk about the treatment dilemma that I mentioned earlier. So our focus now as we move forward is on the clinical area and how we can help in terms of the responder crisis. So really, there's a need to have a big paradigm shift in treatment selection for patients. So essentially, most patients, especially in late stage disease, it remains a major challenge to treat these patients. There are many diagnostic tools available today to guide the use of conventional and emerging oncotherapies, but they lack a strong overall predictive value for the individual patient and often do not translate into effective clinical benefits. So emerging evidence has shown that the entire native tumor microenvironment, not just the tumor, affects how an individual patient will respond to a given treatment. And the spatial distribution of cancer and stromal cells in this microenvironment can affect how they interact with each other and ultimately how the tumor responds to systemic chemo, targeted agents, or immunotherapies. So approaches that focus on simply genes or tumor cells are not able to capture the richness of the signaling and interaction of the microenvironment, which limits their ability to effectively predict treatment. So there's a big market opportunity in colorectal cancer. Just as one example, over 9% of all cancers worldwide are colorectal, and it's the second most common cause of cancer-related deaths in the US, and is expected to increase dramatically worldwide by about 70% by the World Health Organization. A big problem is that today there's at least 9 approved therapeutic interventions in the US, but there is no way to identify for an individual patient which is the best option to treat them. So many patients go through multiple rounds of different therapies. And one of the big questions is, how can we help patients to avoid futile therapies? Similarly, in immuno-oncology, Keytruda or pembrolizumab is the gold standard. And potentially, the potential treatment is around 20% of cancer patients, of which about 10 to 20% are unlikely to respond. So how do we help those patients who won't benefit from checkpoint inhibitors? And how do we avoid treating patients with checkpoint inhibitors who are unlikely to respond? So Neologian's 3D platform really enables personalized treatment. We can test multiple therapeutics in each patient's tumor, and in 5 days, provide the data necessary to guide oncologists about treatment options. So all of our assays are basically functional readouts. And what this enables us to do is give accurate data that relates to therapeutic penetration of tumor tissues and kill tumors, and also do things like identify the correct dosing for a patient. So essentially, we enable a comparison of multiple therapeutic options for a patient. So in the next year, we'll be initiating a couple of clinical studies focused on assessing patient response to treatment using 3D Explore. And next year, we're looking to create a new business entity, which essentially will license Neologian's technology, facilities, and expertise, and apply these to developing a functional personalized test for patients to identify the optimal therapeutic intervention for each patient. And we'll be looking to seek investment partners to develop the business, both as a global entity for personalized care and in the US and in China. And I'd like to thank you at that point and take any questions that people may have.
Thanks, John. That's a really nice presentation. I think there are already a couple of questions from the audience. Let's go with the order. The first one, I think, is from Michael. Michael, you're allowed to talk right now. You can unmute yourself and speak directly. Oh, Michael. You raised the hand, and right now you should be able to talk if you want. If not, we are going to the next one. I don't know your name. It says Huawei. Huawei Mate 40E. You're also allowed to talk. You just need to unmute yourself and speak. No? Okay, then we just read the questions from the audience directly. So the first question is, let me see. It's asking the service is available in China, given that a sample is collected in the United States. Curious about the policy limitation here. So the samples are all from the United States. So the data can cross a board, or the sample can cross board. I think the question is about the policies as well. Thank you.
Sure. So, I mean, I'm not completely familiar with the policies in China, but I do know there's tremendous challenges in terms of working with patient samples in China. And that's only got worse more in the past few years, from my understanding. Certainly in terms of the data we generate in the US for our existing clients, we can provide that data worldwide. There's no limitation from that perspective. I think working with patients in China, that's something we're going to need to look into, and probably we're going to need to set up a Chinese entity that will be involved in receiving and working with patient samples in China only.
Okay, Michael, I'm gonna ask you to unmute yourself. You can see the button. if you see that, you can click in and talk directly. Thanks. And there's another question from the chat box that's asking, can you explain what's the differences between the tumoroid model and organoid model?
Sure, no problem. So organoids, basically, the way you create an organoid is you take patient tissue, you basically break it up, you try and create reserve the stem cells or the cells which will proliferate, you try and grow them again under particular conditions, and then you try and reassemble. So really, they're nothing like the original patient tumor tissue and all the complexity. They don't have stromal elements, they have very limited cell numbers, and they certainly don't have the extracellular matrix. So they really don't reflect the original patient sample, whereas a tumoroid does, because it's all we do is simply physically break up the tumor into these small tumoroids, and then we build essentially a population of tumoroids, which we then screen.
Yeah, I want to ask a follow-up question for that. So it's all of the sample from the hospital, right? So is it possible that the patient can have their own IP, either the real tumor tissue to provide you, and you can do the precision medicine to see which treatment is best and how to proceed with the best therapeutics? Is it possible, or it's like general, general samples for all of the patient populations? Thank you.
Sure. So obviously the principle is for the future, that's exactly where we're going to go, but that obviously has to be done in conjunction with the patient's oncologist. You can't just go and take your own sample and send it in the post. We need to receive patient tissue in our facilities within 24 hours that either biopsies or resections have been taken. So yes, you do need your experts to help you there and give you advice, but certainly we can take tissue directly from patients in terms of the surgical procedures that are used to remove that. It's all the interventional radiologists that take those samples.
Okay. If you have any other questions, please send email to me or to John directly, and we can talk about it later. I think there are still some questions. So thanks, John. It's a really nice talk. It's a wonderful talk, and have a good evening.
Thank you. Thanks for your time.
Thank you. The fourth company. Oh yeah. So we. Yeah, the time is a little bit. It's fine. So the fourth company we have is 13 Biotech. 13 Biotech redesigns drug discovery with biology-driven artificial intelligence to uncover new insights. Its platform predicts new potential therapeutics and pinpoints and mechanisms driving efficacy. It also integrated over 30 types of chemical, biological, and clinical data for each test. Tonight, the CEO and founder, Bradley Pride, is going to talk about it. Brad, you here?
I'm here.
You can go ahead and share your screen.
All right, here we go.
Thank you.
A little bit. So as I'm pulling up my screen, just want to say thanks everybody for coming along, and thanks for having me. I know it's late over here in the East Coast of America, about 9:30, but early morning China. But nonetheless, I'm glad we're able to connect and in all our respective time zones. So can everybody see my screen now?
Yes, perfect.
Awesome. Thanks, Mike. All right, cool. So I think Mike gave a good introduction to what we do. I think AI is very much a black box to a lot of different people. So hopefully I can help simplify that. But as Michael said, there's a challenge for everybody in this industry. That's a gigantic failure rate in bringing drugs from conception to market, about 96%. And we do have a stronghold in oncology, but we do go into different diseases. But with a failure rate of 96% and a disease group like oncology, that means 9.6 million people are dying every year. That was as of 2019. That's very much our mission. What we're trying to solve is not just oncology, but also other deadly diseases, and using artificial intelligence applied to drug discovery to decrease that failure rate. So first, what I'd love to do is introduce my team. we're a group of scientists and then myself, entrepreneurs, which are really hell-bent on this mission to improve that failure rate. We have Neil, who's our CEO, co-founder, has been identified as top 100 AI leader in drug discovery. Olivier Elemento is our scientific co-founder. He actually heads up the largest precision medicine institute in the United States, where 1-3 was born out of. Corey, who's our chief data scientist. She's a background in applied machine learning, and we actually stole her over from an AI hedge fund. So really good find there. Myself, I have a different background for my more scientific oriented co-founders. I was in finance when I started my career in London, moved over into trading. And it was actually after I lost a family member to cancer that I started to move into this industry. I was successful in my first entrepreneurial endeavor where I sold a company in London as the CEO, and then came to America in New York City, where I was really applying my experience building out tech subdivisions for Fortune 500 companies, and really looking for that way, broadly speaking, to get more involved in the fight against cancer, where technology could be applied productively. And that's when I came across my co-founders. By that time, 2018, we were about 7 years into the research and development of technology and started applying it in several different use cases and shown that we had great success. And since we were gone together, we continued with other partnerships, and even to the extent we're building out our own internal programs. So I can talk a little bit about our timeline on the next slide here. So as I mentioned, 2011 was where 1-3 really spun out of the largest precision medicine institute in the United States. And the benefit of this is we're building a series of predictive technologies. So the database itself, and then what the database filters into, which is the algorithms. And we're building that out in a lab. So we're able to make predictions and then see those predictions be actualized or not, and then adjust our technology accordingly. So over the years, we did this within our own lab until we gained the confidence in 2015 to reach out and start to establish partnerships with pharma to further build out different programs and also raise our first round of funding. By 2018, we had some really great breakthrough validations with different pharmaceutical companies where we made predictions such as this drug, this therapeutic will be efficacious against this cancer type based on biomarkers within the patients, the actual target that it's hitting, and other things such as combination synergies. And we made these breakthroughs with AstraZeneca, Oncosystems, as well as several others that you see here today on the screen. And in 2019, towards the end, we raised our most recent multi-million-dollar round of funding and won several awards. We won the Reuters Entrepreneurship Award, won the US-China Innovation Alliance Most Innovative Technology, and started to grow out our partnerships throughout 2020. Had some great breakthroughs as well. So 2021 for us has been continuing those partnerships, releasing the results as and when they're available. But also we started to develop our own internal programs. And the way we approach drug discovery is with biology first. So I think I mentioned it in the intro. What we're doing is we're predicting what is going to happen within the underlying biology in drug discovery. So for us, that means starting first with a target, linking that target to an indication, identifying the ideal patient population, and then repositioning drugs where available towards those targets, or just outsourcing the chemistry. So let me spend the next 2 slides talking more about the technology itself. As I mentioned, I understand that AI can come off as a black box. So just talking about some key components. So when we were in the lab at Weill Cornell, one of the missions for us is really thinking about how are we going to be able to accurately predict biology, one of the most complex fields known to man. And we realized with the general hypothesis in first, which became in practice and became true, is that in order to do that, simply stated, hard to do, but in order to do that, what we need to do is incorporate a multitude of different data types and analyze those in tandem. So what you're seeing on this page here on the chart on the left, very simple chart to understand and see, is that accuracy in predictions of drug discovery doesn't come from a multitude of different data points. It's the data types that you're able to incorporate and analyze together. So that was our first mission when building out 1-3, was actually bringing data together to the point that you can actually combine it for analysis and ingest in machine learning modules. So we first started building out the 1-3 data mapping engine, which is our database. It's incredibly large. It has about 40. Now we have a very fast working engineering team, so the slide's a little bit outdated, but over 50 different biological, clinical, chemical data types from over 200 different sources. To give an example of what could be in there, it's over 100 million different compounds, 66,000 samples, 60,000 targets, and much more. And coming from really two key categories. So private data, where we've acquired this data from nonprofits or previous pharmaceutical partnerships where they've allowed us to use the data in future analysis, and also the public domain of data. So this is available to anybody, but very hard to determine how to effectively analyze this data and incorporate into the database. And that's a huge engineering uplift that we had. So a very simple way of understanding this and using a direct metaphor of the importance of building out a data engine from public domain data is one that everybody couldn't be familiar with, is the use of Google. So the World Wide Web of information of the internet would be almost impossible to go through with a team of people effectively in the speed that Google can do it using their algorithms and their engineering. And that's something that we've done at 1.3 for public domain data in combination with the private data that we've acquired as well. So that's a very high level overview of the value add we've been able to create with our data and different data sources. The next step to really understand, and this is using one example of our capabilities, identifying gene essentiality. And the current machine learning approaches that we've been able to see is that it's a single model with a high level output, which will answer a simple question. So using an example of, is a gene relevant for a specific breast cancer? Now, answering that question, is that or is that not relevant, is incredibly important, but it's very hard to action within the clinic. So what we've done is we've built out a series of different modules, series of different algorithms to tackle the most important question there. If it is relevant, why is it relevant? So if gene A is relevant in breast cancer, is it because it's a protein-protein interaction is activating? Is it because it's synthetically lethal mechanism? Being able to determine that is incredibly essential, and making those predictions so that you can action it with drug discovery is really important. And taking a step back and thinking about what we've created here with our technology and our ability to do what we say we can do and proven out in different case studies, it creates us, it enables us to be end-to-end discovery platform. And in this timeline, which is relatively unconventional to the typical drug discovery methods. First, we start with the target. We identified efficacious targets. We then predict that target's safety. We'll pinpoint optimal patients and biomarkers, and then a match to a chemical compound. And yes, there's also another safety prediction aspect within there too. And I think that's where it's kind of untraditional thing about safety after the target. There's two different elements of predictions of safety, which we'll do. Also going on to see prevalence, to measure the prevalence among the public. And at the end point, what we have is a discovery package with an info on target, drug, and patients ready for development within the clinic. And that's how we partnered. So here on the screen, I've just kind of identified 4 different case studies where we've have quotes from the people who are very satisfied and given an example of what we've done. Given the time that I have left here, I'd love to just kind of grab one of these because I think it still is hard to kind of conceptualize the benefit of drug discovery unless a case study is really walked through. So top left is one of my favorite ones because it has, we're able to talk about it in depth. We're actually going on a different project with this company, Oncosystems, now Termerix, because they've been acquired. And I think it really gives that idea of value that we've been able to generate there from identifying targets, new patients, and then selecting the ideal specific indication. So I'll just jump right into this one. So think about the situation that we had with Oncosystems. When we came across them, they had their drug ONC201. It was initially discovered in a phenotypic screen. So they didn't know the exact mechanism of action, but they did know it was efficacious because they had gone through animal study. And at this point in time, when we met them, they were in a phase 2 trial. And they're doing it in a basket trial against 6 different indication types. And they really weren't seeing that level of efficacy they would like to have seen in human patients. And the objective for us was to first identify the mechanism of action for ONC201 and then go on to pinpoint optimal patients and indications for future trials. The modules that we used in this instance was target deconvolution, gene essentiality, and biomarker identification. So step 1, we needed to identify that ONC201 was a selective DRD2 antagonist. This was a complete novel target at this point in time. And we're using the OTB target deconvolution module. We ranked all different targets, mechanism actions for ONC201. The top rank prediction for ONC201 was, as mentioned, a selective antagonism against DRD2. Again, that was novel, completely new. DRD2 binding prediction was validated through preclinical trials. We then moved on working with Oncosystems since we had that breakthrough and then validated it to determine what the optimal patient population was. So using our gene essentiality module, we ranked 300 different cancer subtypes based on susceptibility to this inhibition, DRD2. So using the OTB biomarker module, we identified biomarkers for top-ranked indications. So there are two, there are glioblastoma and pheochromocytoma. And specifically found that H3K27 methylated glioblastoma was identified as an optimal indication. We have validated this prediction in animals and then worked with Oncosystems to put it into a phase 2 trial. So just completely new phase 2 trial, no longer a basket trial focused on these patients. And for me, this was remarkable to see. So you're seeing massive recession tumors, you're seeing patients' life expectations go up. And you know, as a scientific team, we weren't expecting to be in People magazine, but it's amazing to see these stories from the patients that participated within these trials where you know, mothers and fathers were able to go back to their children and you know, live the lives that they thought they didn't have anymore because it's a very deadly disease. And this is from the humanitarian aspect where there's a huge benefit. But of course, with a breakthrough like this, there is a business aspect as well. There's output left to a $500 million market opportunity and a $400 million cash at $75 million upfront, and then in total other considerations, royalties of a total of a $400 million acquisition for Oncosystems by Chimerix. And we have a quote there with Josh, who we're now working with on another project. We said the platform helps solve problems that they've been working on for years. So it's not just solving the problem itself that is a benefit, it's the amount of time they're able to save with working with us. And now, as mentioned, because we've had successes with our partners and working together on different programs, we've gone in to start developing our own internal programs just because of the commercial aspect of keeping that IP in-house as well. But we are also working with partners. So that's my overview. I know we want to leave some time for questions, so I'll go ahead and take any that people have now.
Thanks, Brett. It's really nice talk. If there are any questions from the audience, you can always raise up your hand. I can unmute yourself, and also you can type your questions in the chat box or in the Q&A session. I can read that for you. Are there any questions? I do have a question for the ONNC 201. And you successfully found the target at DRD2, and it's wonderful discovery. But actually, for the general for target convolution, it's going to be very difficult with a lot of mass spec, and also with a lot of validation with CRISPRs. So how, I mean, what is the way that you just identify the target? Like, I mean, what is the details for you to do that? Because in general, I mean, without even put that into the cells or to the animals, I mean, typically it needs hundreds of hours to do that, right? With the traditional proteomics. So just wanted to ask how you guys manage that. Thank you.
Yeah, so how are we finding our own novel targets for internal development? Yeah, so we primarily and surprisingly, so this is a conversation we've had, and we're kind of going towards, we're entertaining a lot of investment for our A series, right? So it's a question that comes up a lot, and it does take a lot more of a conversation to be able to answer thoroughly. So I'll just scan over, you know, from a high level what we're able to do. So we're taking a massive amount of data. Typically, it's preclinical data. Surprisingly, a lot of people ask the question whether or not this is clinical data and this is knowing targets. These are targets that are completely novel. And then what we'll do is, you know, actually go into early stages of identifying whether or not these targets are efficacious within, you know, in vitro studies. So I think when we do disclose the targets, which obviously we can't discuss for clear reasons, it's a surprise to a lot of people that these could be targets that are potentially efficacious, but it's coming from our database, and it's not necessarily that this literature that says this is a target for this specific indication. It's brand new until tested and then proven out for us. Now with, you know, Oncosystems specifically, this is a brand new and novel mechanism of action which they're targeting. But the way in which we did it, we didn't necessarily need to know that their drug was targeting this first. It can be done in the reverse as well. So not sure if that's going to have a breakthrough moment exactly what we do, but if, you know, someone is interested in, you know, diving deeper in how we are finding this, probably CDA would be required, but feel free to reach out and we can discuss further for whatever purpose the interest is.
Yeah, it's a great finding. It's actually going to help a lot on the targeted convolution in terms of time consuming, energy consuming, a lot.
Absolutely.
Okay, we have another question from the audience. It's asking what database you're using for AI calculation.
Mhm. Yeah, so that was something I was talking about over that slide. So our database is a very large database, and it's coming from over 200 different sources. So really, it is the database which we're using is 1.3 Biotech's database. And that's compiled, so just kind of repeat what was on that slide. It's compiled of private sources, so coming from different pharmaceutical companies established partnerships with, nonprofits, but then also public domain data, and there's just numerous of different places where that's coming from as well. So someone that people would be familiar with, Drug Bank, PubMed, tons of different literatures, so record-leading literatures as well as databases as well.
Thank you. Another question is from Wei Li. It's asking in general how to ensure the quality of the data sources.
Yeah, so there's a series of different quality checks that are beyond me. So this is a data engineering team, and they've put in a series of things. So who leads our data engineering team was someone who was very high up in a financial company where the quality checks that they've gone through is at a magnitude processing. So it's a combination of different automated checks, but then also there are checkpoints for humans to intervene and ensure that this is going through in combination with the computational biologist team, so our R&D team, which will go through and make sure that there's a certain level of scientific rigor. So then going beyond, so that's just bringing the data inwards, going beyond that to make sure that our predictions make sense. This is a huge project, right? Because we come up with such novel targets, there's always going to be a crazy amount of questions of whether or not this could be true before we actually start pushing this into development, because it's extremely costly. So that's, I would say, another kind of endpoint check of the output of our data as well. So it goes on from the actual absorption of the data to the point of running quality checks on the output from a scientific perspective as well.
Thank you. Due to the time, we probably only have 3 questions. It's fine. And if you have any other questions, you can always contact us so we can talk privately. So thanks, Brett. That's a really nice talk. And yeah, if you guys have any questions, please contact us. So thanks, Brad. Have a nice evening.
It was a pleasure. And if there are any other questions, it's brad@13.bio. And thanks a lot, Michael.
Thank you. Thank you.
Take care.
Okay. So for tonight, we in total have 7 companies. So for my part, it's finished. I'm gonna hand it over to Lin. So please don't go away. There are a lot of great companies later coming up. Thank you. Lin, the floor is yours.
Yep, thank you, Michael. And okay, and so from our side, we have 3 companies, and we're gonna start from the Econ Pharmaceuticals, and we will have the speaker Xu Dongyuan, who's the founder of the pharmaceutical, and he will introduce their drug delivery system for the small interfering RNA and messenger RNA. Dr. Yuan.
Yeah, thank you, Lin. Can you see my slide?
Yes.
Okay, great. Yeah, at first, I'd like to take this opportunity to thank for the organizer for this nice opportunity. Next, I'd like to introduce to the company. Our company is called Econ Pharma. We register in Delaware.
It's kind of quiet. Do you mind adjust your mic to make it louder?
Okay, I'll speak louder and hope it's work better. Can you hear me better?
Yeah, I think right now, yeah, if you can speak even more louder, that would be great. But right now we can also hear you.
Yeah, thank you.
Okay, yeah, I'll try to speak louder. Yeah. So our company is called Econ Pharmaceuticals, and currently our location is in New Jersey, and also we plan to build a site in Shanghai. Actually, right now I'm located in Shanghai right now. So this is our team. I think there's some problem with. Just give me a second. Okay. Yeah, so this is our team. So myself has about 20 years experience in pharma. And I used to work for different companies, for example, like GSK, J&J, and RB. I also worked for some smaller pharma in Now Pharma and also the RVU before I founded Econ Pharmaceuticals. Our CSO Dr. Lin also has 20 years experience in biotech. So she used to work for Vertex and Scripps and a few other companies. And our VP is Nevin. Nevin also have about 20 years experience in pharma. So his expertise mostly in formulation. And we also have Yuxuan, who is in charge of pharmaceutical analysis. And Helen is in charge of quality assurance. And also Yang is in charge of the biology and pharmacology. Jennifer is our BD director. So we have some professors serving in our advisory board. For example, like Dr. Minko, who is a professor from Rutgers University, and Dr. Sava from South University, and Dr. Hu is a professor in pharmacology in Rutgers, New Jersey Cancer Institute. Also, we have several doctors like Dr. Bartenberg and Dr. Ball, both from MSK Cancer Center. And currently, we're also working with Dr. Li in the medical school in University of Miami. So our company, we really use some state-of-art siRNA and drug delivery technology to create innovative pharmaceutical products. So we are currently focusing on two types of product pipelines. So the first one is the siRNA and mRNA therapeutics. So the second type is the 505(b)(2) product. The first product that we are currently working on is for the pulmonary fibrosis. So as we know, there are about 5 million people affected by pulmonary fibrosis globally. However, the treatment option for the RPF is very limited. And currently there are two drugs approved. The first one is pirfenidone. The second one is nintedanib. However, these treatments can only improve the symptoms temporarily, or just slow down the disease progression. I mean, they cannot really cure the disease. So the patient's life expectancy is short. So generally it's about 3 to 5 years. And so therefore, this pulmonary fibrosis is a highly unmet medical needs area. The market currently is estimated is about about 4 to 5 billion. And it's also estimated will grow to add to 9 billion by 2027. So we designed some siRNA targeting to some profibrotic genes in pulmonary fibrosis. For example, like MMP3, CCR12, and HIF1A. And we select the best candidates by using some cell studies. Then we advance the best candidate to the animal study. So as you can see from the left, from the CT image, so the normal mice, the lung has a very nice shape of the lung, the normal, which is very normal. But for the diseased mice, you can see a lot of fibrosis going on, and also the volume has shrinked a lot to about 1/4 of the original volume. The third one is the RPF mice, but is treated with our siRNA lipid nanoparticles. And we use the inhalation method to deliver the siRNA lipid nanoparticles to the mice. As you can see, we can still keep the function of the lung, and also the volume is very similar to the normal lung. And also we look at the survival rate. So for the mice who has the disease, the mice will die very quickly in 2 weeks. But for the mice who is treated with the siRNA lipid nanoparticles or siRNA LNP, the mice can still survive after 2 weeks. So about 80% of mice can survive after 2 weeks. And about 70% of mice can survive after 3 weeks. And for this formulation, we also did some other studies. We did the in vivo gene expression study. So we want to make sure the siRNA delivered by LNP can effectively silence the targeted gene. And we can see this gene expression has been downregulated. So That means that the siRNA is working properly, in the animal model. And we also look at the side effects like body mass and lung mass. So we did not observe very significant side effects. We also look at the biomarker like hydroxyproline to look at the fibrosis level. So once we use the siRNA LNP, so we can observe that the gene silencing level or the collagen level or the fibrosis level has been significantly decreased. We also did some pathology studies, and as we can see, for the RPF mice, the lung has been damaged, and you do not see much of the normal structure. But for the RPF mice, which is treated by siRNA LNP, so we can still maintain very good the lung structure, very similar to the normal lung tissue in the mice. We also did the distribution study. So for the siRNA LNP with inhaled or by using the inhalation method, we use a nebulizer to help to deliver the siRNA LNP into the lung. And we found that about 80% of the siRNA LNP nanoparticles are deposited in the lung. And about 7% go to liver, 9% to the kidney, but the majority is in the lung. But if we use the RV method, we can see that the majority will go to the liver, 39%, and 41% will go to kidney. So only 6% will go to the lung. So therefore, by using inhalation method, that will be the best delivery method for treatment of pulmonary fibrosis. The other study we're currently doing, we also use the siRNA LNP for the treatment of COVID-19. So we're not developing a vaccine, but instead we're using siRNA as a treatment. So basically, we use the siRNA to target the mRNA of the COVID-19 virus. Basically, we use the siRNA targeted to some conserved region of the virus. So in that way, we can deal with the different variants. And after we bind the siRNA with the targeted mRNA in the virus, so the siRNA can silence the mRNA and degrade the mRNA. So therefore, we can prevent its replication and treat the COVID-19 infection. Apart from those siRNA LNP projects we are doing, we are also developing another product, which is a long-acting antipsychotics, Macrosphere injection, and for the treatment of schizophrenia. So as we know that patients need to take this medication every day over long treatment period for the schizophrenia, bipolar disease, and depression. But for the long-acting antipsychotic Macrosphere injection, so we only just need to inject once in a week, or maybe once once in a month, or maybe once in every 3 months. So which is very convenient for the patients. So otherwise, the patient need to take the tablets or capsules every day, or maybe 2 times or 3 times a day. As we know, the schizophrenia drug market is also very large. So it's may reach about $9 billion by 2026. Basically, what we did, we load one small molecule drug for the treatment of schizophrenia into a Macrosphere. So the Macrosphere is made of some biodegradable polymers. So after the water enter into the Macrospheres, it will degrade the polymer. And then the drug will be released very slowly over a period of time. Can be as long as 1 month or maybe 3 months, depending on the design of our Macrospheres. So then in this way, it's very convenient for the patients, and very convenient for their doctors, and very convenient for their families. So basically, the patient just need to take 1 injection of these long-acting Macrospheres. then he doesn't need to worry about medication in the next 1-2 months. So it's which will give a very good patient compliance. And for this product, we also applied the patent. Currently, we have the PCT patents. And also for the first product that I mentioned earlier for the RPF, SIN-LNP, we already have the patent granted by the USPTO office. And currently, we are raising $80 million to support R&D-enabling studies and prepare to submit to FDA and CDE in China for the clinical studies. We plan to advance the SIN-LNP inhalation project to the phase 1 in the next year, and also the long-acting Macrosphere injectable project to the phase 2 in next year. Because it is for the long-acting Macrosphere is 505(b)(2), so we can request a waiver, for the phase 1, which is a safety study, and go directly to the phase 2. And some other costs, some other fundings we're going to use for the daily operations for this year and next year. And this is my contact information. So if you have any question, and if you are interested in collaboration with our company, please just reach out. And also, this is my WeChat, and please feel free to add my WeChat, and we can communicate anytime. Yeah, thank you very much.
Okay, thank you, Dr. Yuan. And if the audience have any questions, please raise your hand, and we can unmute you to speak. And also, feel free to use the chat box or Q&A box to send a text message. Yeah. Maybe I'll start with one question. So for the in one of your slides, you show that the inhalation method can help us to avoid the accumulation of LNP in liver. And what about the efficiency? Does it also comparable with the injection method?
Yes, I think you're talking about about this one. Yeah. The efficiency actually would work better for the injection, because the injection, because you can only have 6% will go to the lung, but by using inhalation, you will have as high as 80%. But just for the amount that enter into the lung, let's say if you 6% in the lung, and I also use 6%, if you have 6% of the lung by injection and also have 6% by inhalation, they will work in the same way. Yeah, but just using inhalation, we can deliver much more of the dose directly to the lung. And there's some benefits for that, because if we use inhalation, so it's the drug will deliver direct to the lung, we can avoid the liver first pass effect. If you use injection, because of a liver first pass effect, a lot of drug will be metabolized in the liver and cannot deliver to the target site.
Yep, thank you. And we have a question from the audience through the Q&A box. So the question is from Tony. What's the difference of Econ's LNP in regards to the incumbent MC3?
Yes, so our LNP, basically we use the different cationic lipids or ionizable lipids, because MC3 currently is used in the vaccine, the COVID vaccine, LNP. For example, could be in Pfizer or in Moderna's lipids, nanoparticles, but we use a different lipids. So in terms of the patent and IP, we have no issue, conflict with their patents. And in fact, so our AMP patent has already been granted by the USPTO office.
Okay, great, thanks. Yeah, it's good to know. I think that's also answered the second question regarding to the patent of the LNP.
Yes.
Yep. Do we have more questions from the audience? Okay, there's one more. Can you share more information about the preparation for the production and quality control, such as the composition of the LNP, and will Icon do the production by yourself?
Yes, yeah, that's that's a good question. Yeah, so basically, we prepare the production by ourselves. We manufacture the LNP lipid nanoparticles by ourselves because I mean it's very difficult to find a CMO can do the LNP for you, and also be nice to keep all the confidential information and also commercial secrets in house. So basically, yeah, we do the production by ourselves. And in terms of the method of preparation or the preparation for production, yeah, so we use I would say similar methods as Moderna and also Pfizer. We use a similar LNP production methods. The quality control is basically, so you need to characterize, for example, the particle size, the zeta potential, the drug loading, the stability, and those like quality parameters. So you have to control them. I mean, the composition is similar to the vaccine LNP. So basically, you need to have cholesterol lipids, helper lipids, cationic lipids. These are several ingredients. In some other LNP, so they're very similar. And for the macrosphere, basically, we have the drug itself, which is a small molecule, and for the polymers itself, so we use some biodegradable polymers in the macrosphere, and once it's degraded, so there's those components, monomers can be easily absorbed by the human body, so there's no side effects at all, very safe. And if you are interested to learn more, so please just feel free to contact with me so we can discuss this offline.
Yeah, great, thank you. And very precise time control. And nice talk. And if the audience has further questions, please reach out. And our next speaker is from Accelements Corporation, and the Dr. Ching Wu, and I think we will have the introduction about a novel spectrometry design and related AI software. So Dr. Wu, can you share your screen? Okay.
Yeah, great. And can you guys see the screen?
Yes.
Oh, thank you. Thank you for the opportunity to speak here. And again, my name is Ching Wu. I'm the president and CEO of Accelements Corporation. I have a more descriptive title for you guys. And so what do we do? And we are a design and manufacturer of mass spectrometers. Today, I'm going to talk about accurate measurement of chemical and biologics at the point of needs. So there's a couple of keywords, and we want to bring the mass spectrometer outside the laboratory, and that's probably one of our innovative part, and that's also in the entire the technology development from the beginning of the company, and we also bring the accuracy to the field. So that's probably 2 points I want to emphasize. And of course, I'm going to move on and continue to talk about the specifics. So with the platform technology of a mass spectrometer, there's like a broader market we're addressing, and you can probably see those systems in the homeland security area. When you go through the airport, there's a device screening explosives and make sure you are safe. Under environmental monitoring devices, they're like a smaller portable device, and some of those are mass spectrometers. And they're like a food and drug and medical diagnostics. there's like a huge market, and there are high demand of a high accuracy, robust, high performance system that give you accurate information. And we talk about the AI, and lots of those, data need to be acquired, and then we can apply our data algorithm to extract the true information out. And so the accurate high performance measurement is the key. Today, I'm going to focus on a couple different aspects. One is the drug manufacturing process, which is a life science tool we probably see ourselves in. And another area is a medical diagnostic area. We are trying to bring the mass spectrometer as a point of care measurement device for medical diagnostic. So what problem we're solving here is the traditional mass spectrometer that have been used for decades is very much a laboratory device. It's you have to bring your sample to the device. It normally occupy half of your room, the entire bench, and that require a very skillful, person to operate. So for this device to be able to use in the field where the chemical measurement is needed, is very challenging. So on the other hand, you probably have a simpler device, a portable, so-called portable mass spectrometers, or a mobility spectrometer, or chemical sensors. And those are all suffer a common problem. They are not accurate enough. So how do we bring this accurate golden standard mass spectrometer into the chemical measurement is needed, right? So that is where our effort starts. So we very much, not only shrink the size of the mass spectrometer and the portability, and we also, make it more accurate and in a smaller form factor. When you operate this system in the field or wherever the point of need, you will need the device to give you a quick answer. It's no longer you're running a long laboratory procedure and take hours or minutes to do that, and sometimes they require you to get answer in seconds. So that is the speed required. And also, often when you bring them outside the lab, you no longer have a PhD scientist to operating them, and you need to make that non-expert user friendly. That's easy to use, right? So there's this 4 different aspects we are delivering our device. So the key part of this innovation or improvement is the high performance mobility spectrometer. So you're probably not very familiar with that term, mobility spectrometer, but you're really familiar with the airport screening system. Every time you walk through the airport, you'll be screened with a device. When they collect a swab sample from your bag or sometimes from your hand, they will put it into a device, and then quickly they have a readout whether or not you are exposed to any explosives or controlled substance they care about. They will stop you or let you go, right? So that is a typical operation of a point of need chemical detection device. What we did, we make that device a much higher performance and comparable to the laboratory HPLC performance. Now we can make this device to be used as a life science tool to do biologic sample and chemical samples we carried in the life science and other research area. Yeah. So this device, there's improvement of the high performance mobility spectrometer, and it can be used still in the laboratory. And we have a collaborative project. You can see Thermo is co-marketing with us, and Thermo Fisher is one of the major instrument company in the field. And we have a co-marketing agreement with them where we put our improved mobility spectrometer for the laboratory performance. And we also have our improved mobility spectrometer go back to the security area. And if you probably can see the other brand of mobility spectrometer, but they have excellence inside. And all this evidence basically just marking our technology and our effort in this development of the technology. We have 50 some patents and probably have 20 some $million invest in the R&D, and we are producing this device now. So combining this high performance mobility spectrometer with a mass spectrometer open up a whole new applications. So that is our latest device, and this is our major effort today. So we are proposing to use this mobility spectrometer, mass spectrometer in the medical diagnostic area. Imagine, the measurement you normally have to send to the laboratory, and now you can do it at point of care, next to wherever the measurement is needed, like emergency room. Those kind of examples, numerous examples, you need a faster, easy to use, high accurate instrument. In principle, and we have a better device. And traditional mass spectrometers are only measuring the molecular weight, right? So you know the weight, you know what kind of molecule they are. But we're not only doing that. With the mobility measurement, we can measure the molecular structure. So you have a structure and weight measurement. Now you have a better description of this particular molecule. And so if you're talking about a molecular ID, this two-dimensional device is going to give you a better answer. Compared to all the traditional systems, and I probably don't want to repeat all the scientific terms, you'll get a better speed and accuracy, resolution, sensitivities. And not only that, like I mentioned, the airport system. So the mobility spectrometer have a nature can be used in the field. So when you use in the medical office and you share the same nature, now you have a device that can operate a long hour without a maintenance, and it's resist to the contaminations and the harsh operating environment. Those are key factors for the system to get out of the laboratory. I'll give you a couple of case studies we're doing today. And so one is what we're selling a lot in the pharmaceutical manufacturing process, this particular application called the pharmaceutical cleaning validation. Basically what it is when you're manufacturing a batch of drugs, and normally what you want to switch between one drug to another, normally the equipment need to be cleaned. When you're cleaned, you got to make sure, report to FDA, you do have a clean device, right? So they sample from the surface and prepare the sample, put it into HPLC, make sure certain APIs is lower than the level required. So this process may take a day, may take 2 days. And when the manufacturing shut down for a day, they may lose $60,000, they may lose $1 million, right? So if you put a sample in our device, you can do that at line. You can immediately tell whether or not the contamination is there. That's almost resemble to airport applications, right? So, and you can also put a batch of this sample into an auto sampler and get this thing done maybe within an hour or two. So that gonna give you a shutdown, much shorter shutdown time, improve the efficiency, right? So, some of our customers basically saying, okay, well, we're buying the equipment, my ROI is only a month, right? So, they will get their money back really quickly. That's probably the customers that you're looking at, major pharmaceutical areas, and there's like a very significant feedbacks, and the people are looking at it. Doing the cleaning validation effectively will give you will allow the pharmaceutical company to save money and improve their efficiency. Another example, and I want to emphasize on this, and we are trying to bring that mobility spectrometer, mass spectrometer technology into the clinical area. That is going to be a point of care system. Imagine you are using a mass spectrometer like this setting. You need a PhD scientist and operating for minutes or hours to get a result. You can put a sample in, get a quick readout, right? So the speed is tremendous. 20 milliseconds, you get the same result as the traditional HPLC for 20 minutes, right? So 20 milliseconds is like a fraction of a second. You can just do the measurement quickly. And we have a plan to move into China, and we are talking to the local governments, and we try to, kind of get a manufacturing process going on there. And so we will be making manufacturing the system there. So probably go to China. Another reason is we can quickly get approval, for medical applications. So this is probably one of the major efforts we are doing. Compared to all this instrument in the market, and we have clear advantage over the others. And if you are familiar with you are in Boston area, you probably see that the recent IPO of NIOA devices. They have a similar kind of approach, but they don't have the performance. That's the one of the system what I was talking about. Have the accuracy in the field is most important. We will compare to them. We probably gonna beat them from the performance point of view, but we'll share the same market, right? So in-field mass spectrometer is the market I'm trying to approach to. Compared to the laboratory system, we have very similar performance to the laboratory system, but we have the portability I've been talking about. The market size, we believe we can address about $2.4 billion market, including the trace detection, clinical validation, clinical mass spec area. And with the development, eventually we probably can reach a much greater market size. So we are currently working on the pharmaceutical manufacturing, and including a biopharma process, point of care system. And we are partnered with somebody else working in the security area. So our own focus is only the life science tool and the point of care system. We have a complete team, and including myself as a technology leader at GE Security before I start Excellence. Mark Osgood is a former mass spec engineer from Bruker Electronics, if you're familiar with that. And so probably that's one of the leading instrument manufacturer. So this round, I will try to raise $10 million and to complete our management team, and we try to bring the revenue next year to a significant increase. And the more important, we try to set up a operation in China and try to scale up our manufacturing. At the most critical aspect of this is to get certification approval for the point of care mass spec system. So that probably where we're going to use the money. To summarize what I've been talking about, and so we're directly addressing a $1 billion market, and we have significant technology advancement, 50-some patent, multiple international awards. We have a management team, and we already deployed over 100 units in the market. So that probably give you a clear profile of the size of the company. As a bonus, we're developing a triple quadruple mass spectrometer. We have the team and the international experts in mass spectrometry around us, and we are developing next generation mass spectrometers. If you're familiar with the clinical mass spec area, you probably know what I'm talking about. A triple quadruple mass spectrometer is hard to do, but we do have the original inventor on our team, and we will address that issue. So I should stop here, and I probably use up most of my time. And thank you very much for listening, and I'm ready for your questions.
Okay, thank you, Ching. Very interesting device. I think we already have one question from the audience in the Q&A tab. It's from Li Bingcui. He's asking, could it be used for COVID-19 test? What is the speed and the cost?
Yeah, that's a great question. I did not get into that. I do have a slide, and we did a quick, very rather novel test on the metabolites in your urine, and that correlated to the COVID patients. We tested about 800 patients, and get a sensitivity about 98%, and we do have a specificity like 88%. It was a little bit lower, and because our sample size was relatively small on the healthy people than the patient. So we do believe that gave us a clear indication, and we did not make enough effort to make it commercialized, and but we do look at in the future and on a similar disease. If you have a virus infection, a virus in your body, it will change your metabolism, and you will see different patterns in the metabolites. So for this COVID case, and we did submit to the FDA for the pre-EUA submission. So because the situation go up and down, and as a business, we did not fully pursue that opportunity, but give you a flavor of it. So.
Yeah, thanks. Yeah, if the audience has more questions, feel free to raise your hand or type in through the Q&A and chat box. Meantime, I have a question. So we mentioned the word sensitivity multiple times. I'm wondering that if we perform the untargeted proteomics on this new device, like how many proteins can we detect from the human sample in general?
Oh, we didn't do those kind of study, but the traditional you're more hitting on the original principle of if you use MS only a device, you use an Orbitrap or whatever the highest resolving power mass spectrometer, you may see X number, and maybe I don't remember those numbers, but probably thousands of those proteins, and if you use mobility with a mass spectrometer, and so you will get a lot of more IDs. There are papers, and probably if you're looking for mobility mass spectrometry, you will get a lot of those answers. And I don't have a specific number for you, and so probably we can look it up.
Mm-hmm. I see. Yep. Thanks. Are there more questions from the audience? Okay. If not, let's thanks Ching again, and thanks for the nice talk. And
thank you.
Yeah.
Thank you.
Night. Like if the audience has more questions, feel free to connect. Thanks. Then our next speaker is Dr. Irene Tong from the Radium Bioscience, and they're also focusing on the mRNA-related therapy. And Irene, you here? Irene, you are on mute.
Okay, great. Can you hear me?
Yes. You want me to share the slides for you?
Yeah, that'll be great. I appreciate it.
Okay, let me see.
Okay, great. Thank you very much. Thank you everyone for joining this meeting, and it's a great honor to be invited to give a presentation here. So I'm just gonna give you some introductions of what the work we've been doing and the MI research we are focusing on, and hope that could help us to get to know each other. So basically, I just wanted to start with a little bit background of what MI is and how important the research is, and why do we want to focus on MI research. So I think most people get to know MI research due to the pandemic, because MI vaccines works well in the pandemic. And we do know like the large companies like Moderna, BioNTech, and CureVac, they're all doing very well in the pandemic, and the MI vaccine works very well in COVID-19. Well, actually, MI research started many years ago, and people has been trying to work on MI as a good research tool to do both vaccine and medicine for many years. And the big challenge is the stability in the delivery of MI into a cell, and how can we stabilize MI into a cell, whereas it also has a large duration of the time. So basically, MI can work as both as vaccine and the drug. So how we start this? Can you hear me clearly?
Yes, we can hear you. Can you see the slide?
Great.
Like, let me know if you want to move to the next page.
Yes. Yeah. Okay. Sure. So I can see the slides quite quickly. So basically, we started this idea when I was first came back to China, and that's actually for before the pandemic, and we do see the growth of the mRNA companies, and we think it's a very good tool for both vaccines and also both as medicine. So then I start to talk to ex-colleagues in Moderna to see whether people who are interested to start a new mRNA company and try to dive into new areas, so we can actually see how mRNA can work on many other diseases as well. So basically, most of the team we have now are ex-alumni in Moderna, and I started to do this 2 years ago. That's before the pandemic. So, and at that time, LNP was worked out in Moderna, and the vaccine was working well, and people started to try to work on cancer vaccines, into cancer disease areas and rare diseases and chronic diseases at that time. So then I started to try to get people out of Moderna to start a new company at that time, and we built up the Boston team. Could you go to the next slide, please? So then later I started to do roadshows in China, because I wanted to do a center both in China and the United States, and to try to find the experts in China so we can team up to work on MI as well. And could you go to the next slide, please? Yes, so this just shows the history of mRNA vaccine. So you can see people started to work on this actually almost 100 years ago. And the largest challenge for mRNA as a vaccine and as a drug is its stability and the delivery issue, because mRNA is very unstable. And at that time, Dr. Rob Langer, well, he's a pioneer in mRNA vaccine and as well as mRNA delivery. And actually before that, both Elena and Sarepta, they were working on siRNAs, and those are the delivery system we use in like GalNAc, and also the ASO platform, because I had an ASO platform company in Hangzhou when I first came back to China to do ASO company. So all these drug delivery tools, they were trying to stabilize either mRNA or siRNA, because siRNA also has stability issues, and also it's very toxic compared to mRNA. Because mRNA is hitting the central dogma, so it's less toxic than siRNA, so that's its advantage compared to siRNA as vaccine tools and as drug tools. So the traditional way of doing mRNA as a vaccine, it took people many years to develop the LNP system. It was actually originally from Dr. Langer, and he licensed out to two companies. One is one company in Canada called Genevant, and the other is a company it's also a Canadian company called Quintus. There are two mRNA CDMOs, and they actually made this LNP platform from academic LNP into a CDMO LNP. Genevant modified the LNP into LPX system, whereas Quintus actually keep this as an LNP platform. And then later, Genevant licensed out this LPX system into BioNTech, and then later into Pfizer, and then into Fusion in China. So that's one part of the story. The delivery system has been modified from LNP into LPX, and you do see like this is how it developed in BioNTech, and then in Pfizer, and then in Fusion in China. Well, actually, it's also getting expedited approval in China as well. And I'm actually now in Beijing, in China, to set up our R&D center in Beijing. So the other branch is actually from Quintus, and it goes to another company called Arbutus. Arbutus is the company actually working on HBV. It's now working on COVID-19, and then they have some legal issues with Moderna due to the lawyers being not very careful about the IPs of Arbutus. So then, but we're not going to talk about that here. So basically, in license to two companies, one is CureVac in Germany, and one is Moderna in Boston. So then CureVac used this LNP to deliver both and to work on both the COVID-19 vaccines as well as its cancer vaccines. And it has lung cancers, those are the large ones in Europe as well as here in China as well. And also Moderna actually developed the Aquantis LNP into a more advanced and industrialized LNP. Actually, it has been internally modified 4 generations within Madara. Madara has a full functional LNP team to improve its efficacy. So then you'll be curious about then, so why CureVac and Madara are actually so different in terms of its market value? Well, actually, it's because of the quality of mRNA. So mRNA also has several generations, and the raw generation, the first generation is called raw mRNA. Basically, it's mRNA itself, and then later, then we have modified mRNA. That's what Madara used. So basically, CureVac used the raw mRNA, whereas Madara used the modified mRNA, and that's why Madara's mRNA is more advanced. And so then you can see from the results from both of the vaccine as well as from the cancer drugs as well. So that's the second generation you call modified mRNA, and then later we have self-amplifying mRNA, and now we have circular mRNA and many more other mRNA platforms, as well as many more mRNAs coming out. Some of them are tested in clinical trials, some of them in labs, some of them in startups like us. So you do see the booming of the mRNA industry globally, or starting in Boston and then to Germany, and now in China it's getting more and more popular in China as well. So could you go to the next slide, please? Yeah, so this shows how it was difficult many years ago, because the entry into the cell as well as stability, and also mRNA has to be recognized, because it has to go to a specific tissue, and it has to be recognized by receptors or ligands in the tissue and in a specific area of the disease. And you also need to think of how can we stabilize it to stay in the specific disease area and to make it more functional. And so those are the challenges for mRNA, both as a vaccine and as a drug many years ago. Could you go to the next slide, please? Okay, so then now, due to the development of the delivery system, it's called LNP, the lipid nanoparticle. It's basically the lipid nanoparticle that can help to stabilize and to deliver the mRNA into specific tissues. Or initially, it was not that tissue specific. It's just basically, sort of like circulate in your body and goes to wherever it could go. So then later stages, people need to develop LNPs that has the tissue specificity and can recognize the ligand and receptors. Initially, it couldn't recognize ligands and receptors, just goes to wherever it goes, and it's sort of like a bad chance. So could you go to the next slide, please? Okay, so this basically shows the process of how mRNA vaccine was made, and so basically initially we need to get the sequence of the virus. So say let's take COVID-19 as example. Actually the sequence has been is already public, so you actually get the sequence. And well, if you're working on other viruses, you don't really know what exact sequence you want to work on. There are AI tools. Well, actually right now, in addition to Madara, Madara is of course the best. In addition to Madara, there are many other companies or people from pure tech background, like here in China, like people working in Baidu, they can develop the AI tools to basically to predict and to find the correct mRNA virus and to optimize the mRNA sequence you wanted to work on. Because if you wanted to synthesize and modify the mRNA sequence, if it's too wrong, or if it's if the sequence is too complicated, it's getting a little bit difficult. So you wanted to find the correct sequence, you wanted to optimize it, and you want to work on it so it's easier for the scientists in the later stage to basically to work on the mRNA sequence. So the first step is actually using AI as a tool to do the screening and to optimize the mRNA sequence that people can work on. So that's the first step. And later step then is sort of like you need to pair the LNP with the mRNA. Due to the length and the structure difference of mRNA, because initially most of the mRNAs will work on a linear mRNA, and now we can come out with circular mRNA, so it's getting more complicated in terms of secondary structure. So basically, you need to kind of like to pair the LNP with correct LNP with a correct mRNA, because LNP is not just one patent or just one delivery liposome. Is actually a mixture of a lot of liposomes, and I can talk about the contents of the LNP later. So you need to pick up the correct LNP, and sometimes you could not find the correct LNP for the correct mRNA, then you need to synthesize a new one. So it actually takes a lot of time and efforts, and need to have people who have very specialized specialty in LNP to work on this. Could you go to next slide, please? Yeah, so this shows how mRNA work as a medicine. So basically, you can see that the cell take up mRNA into the cytosol, and the mRNA get released. So it's very similar to my antigens. Well, if you had experience to work on neoantigens or work on TSAs and PASs, then so you should know that you need to find tumor-specific antigen. So then that's an advantage of mRNA to compare to neoantigens as the new medicines, because you don't have to as PSAs that you could not find the tumor-specific antigen or tumor-specific antigens, because that's like a named antigen, and sometimes you compare the samples of patients and the normal people or people who had the indications but not sick yet, and you do the comparison, and you just don't see any significant difference, or you do see some difference, but they are actually chaos or random mistakes. So in that case, then it's getting a challenge for the neoantigen field to find the exact tumor-specific antigen, as people claimed, was as mRNA. It's sort of like a central dogma. So every mRNA is present already. You just need to compare how the difference, the expression level difference, as well as the presence, and also the duration of the experience. You compare the patient samples with the normal people or people who had the indication but not sick yet. So you understand what exactly mRNA is working on this. And actually, based on the disease biology, we have been studying for hundreds of years. Many of the diseases have known targets, like in tumor, IL-2 or IL-15 or IL-12, or KRAS or OX40. So these are known targets. People know many years. It's been the challenge for drug industries. People have difficulty to actually do the on-target effects using either small molecules or antibodies or other methods or immunotherapies or antigens. People has been working on for mRNA, as well as you can synthesize the mRNA in a good size and also in a good quality and good purity. Well, that's actually a challenge for many years, but now it's getting solved step by step. Then you can have the mRNA to work as a new class of medicine, and you do have it's central dogma, so you do have the sequence being present already. So that's sort of like an advantage for mRNA as medicine compared to traditional medicines. Could you go to the next slide, please? Okay, so this is basically showing how the process, how mRNA is working as a medicine, and I just explained why it's better than neoantigens or antibodies, because its central dogma is present already. So you have the sequence already, but the challenge comes out: how can you get the correct sequence, and how can you optimize the sequence, and how can you find the exact sequence that's been different from the patient, the carrier, and the normal people. And do these carriers really represent the real disease biology, and do this being consistent with the traditional disease biology people has been studying for many years, because those are known knowledges people already know, targets like IL-2, IL-15 I've been talking about. So you need to make sure it's consistent with existing knowledge, and you also need to make sure that the new ones coming out do have a disease biology that could be explained, not just chaos or mistakes from the machine or from the AI. So could you go to the next slide, please?
Yeah, Irene, just want to remind you, if you could wrap it up like in next 3 to 5 minutes, then we can still leave some time for the audience.
Okay, great. So basically what we've been, so I'm going to skip the team part. So basically we have a team from Madara, and also we have a team of experts in China as well. And could you go to the next slide? So basically right now what we're doing is we set up the LNP library to do large scale screening to find the LNPs that's been tissue specific and target specific. We'll go to the next slides. And tissue specific, target specific, less toxic, and also general, which means one LNP. Could go to the next slide, please. Next slide. So these are the team, people from Moderna, and these are the team, what the Chinese experts we have now. Go to next slide. Next. Okay, I'm gonna skip the team part. Go to next. Yeah, so this is what we've done. Go to next one. So basically, we have international PCT. We have patents both in China, United States, and Japan, and Korea. All the large markets globally. Can go to next slide, please. So these are the patents we have now. And okay, go to next slide. I'm just gonna show what exactly we did. So basically, we set up the LNP library. Go to next slide. So these are the patents we have. These are the patents. Go to next slide. Okay, so go to next slide, and this is how we did. So basically, we set up. Go to next slide. This shows how LNP works, and this is the LNP library we built up. So basically, we set up an LNP library to get to the LNPs that's less toxic and more specific and more general, which means it can be tissue specific, target specific, and also less toxic, because the toxicity of LNP comes from the addictive and also some of the catalytic lipids and of the components of LNP. Go to next slide. So this is a library we built, and the MI we use is a self-amplifying MI, as I mentioned last time. It's coming from raw MI CureVac and modified MI Moderna is using, and now we're using self-amplifying MI, which means it can amplify by itself. So in this case, it's less. So it's lower cost and higher efficiency, and also higher penetration of the tissue into different organs, which means you can, go to more disease areas. Go to next slide, please. Okay, so this shows the advantages and disadvantages of LNP. As a measuring tool, LNP has advantages, in terms of high efficiency delivery of MMIs. Well, but it's not a perfect system. It has some disadvantages, some toxicity, as I mentioned, tissue specificity, and some tox, and also in terms of on-target effects, as well as the duration of the drug of MI as a medicine. So this is how we develop. We sort of like advance this from a specific LNP into general LNP, and in this case, we did it from an LNP library. Go to next, and this shows how it works. And this is the data. So basically, using this LNP library, we kind of like develop LNPs that can deliver multi-MIs. In this case, that means it's lower cost and also higher efficiency and less toxic. And also in terms of, if you're moving from a vaccine to the drugs, to medicines, you think of the disease, many of them are coming from multi-synchrony pathways, and it's more complicated than one drug for one pathway, or just single mutations, or like COVID-19, just one RNA viruses. So in this case, we can solve problems in more complicated situations. Go to next slide, please. Okay, so this is basically the data we have, and you can see the delivery efficiency of the LNP library we developed in the mice, and we actually compared our data to Moderna data. Go to the next one. So these are the data from Moderna, and you can do the comparison of our data from to compare to the Moderna data, because we actually built up the LNP library to screen for high efficacy, lower tox, and more general LNPs, and also the LNPs that could deliver multi-MIs. In this case, that means you could be less toxic and high efficacy, as well as to actually go to more disease areas. Could you go to next slide, please? So these are more data from Moderna, and we did a comparison. You can look at the data later to see how our data, compared to the data in Moderna had. Go to the next one. Okay, so these are LNP data. Go to next one. Okay, so these are the old data we had using the ASO platform. So ASO platform is a platform that can dive in, it goes into the brain, so it works well in the brain, rare disease and non-rare diseases. Go to the next one. And this is how we developed the LNP platform. Basically AI design, the formulation, that's manual, and you do the screening to build the library. Go to the next one. So right now, next one, please. So these are the old data we had in the ASO platform, and we're going to skip this to save some time. Go to next one. Okay, this is the old ASO data we had, and go to next one, please. Okay, so right now, so basically we have our Shanghai center. I didn't put the Shanghai one ready here. So we have our Shanghai center ready to go, and I'm setting up the R&D center in Beijing and Boston simultaneously, and we're raising funds both in the United States and China. Go to next one. Right now, so what we are doing is right now, as I said, we are building up this LNP library to screening for higher efficacy, lower tox, more tissue specific, as well as LNPs that delivered multi-MIs at the same time that can solve both linear and circular and self-amplifying MIs in a more complicated MIs. That's what we've done. And I'm raising funds both in the United States and China and to basically to go to next one. Okay, go to next one, please. Okay, so this is setup of the RNA is free lab. Go to next one. Okay, go to next one. So these are the main product we're working on. It's for the mutant of the COVID-19 using mRNA, and we're using self-amplifying mRNA to develop the Delta vaccine. Go to next one. So this is the main product. So these are the go to next one. These are the PDs and go to next one. This is BD model, business model. So these are the pipelines we're working on. So the main product is the COVID-19 Delta vaccine, and for others, we have a bank of both infectious diseases. And go to next one. The next one is mostly oncology, as you can guess. So it's infectious disease and oncology intense. And the main product is the COVID-19 Delta vaccine. And we're using the LNP platform we developed, and we are working on it for the global international PCT. Go to next one, please. Okay, so go to the next one, please. So basically, we're raising funds both in the United States and China to build up the R&D center both in Beijing and in Boston. And we're working on Delta vaccines, and that's the main product we're working on. We actually improved the LNP by the LNP library we developed. And basically, it's higher efficacy, lower tox, more general, and the LNPs can deliver hundreds of MIs at the same time. So in this case, which means higher efficacy right now, our efficacy is 95%, and that's the data we get, latest data we get, and actually I'm very confident with more funding we can actually get better data, because then we can invite more experts, and also we can get more talented people involved in the project, so we can improve the data into a higher efficacy as well. Could you go to the next slide, please? Okay, so this basically shows what we're doing. We're raising funds both in the United States and in China. Right now, I'm in Beijing, and we have people in Boston as well. Go to the next one. And this shows like the plan. So we aim to go public both in NASDAQ and also in China and Hong Kong as well. So in this case, people can we can leverage the resources both on both sides. And for Boston, we have a modern team, so people are very well trained in terms of Mri vaccine science, and I'm actually doing a roadshow, and I'm giving talks online in China as well. So in this case, then we can actually unite all the experts in China as well. Go to the next one. So, and this is the business plan I already mentioned. Could you go to the next one, please? Okay, so if you're interested, if you have expertise in Mri vaccine or medicine or the delivery system, or your researchers, working in the lab, you have publications and patents, please feel free to add my WeChat and join us as a team. Or if you're interested in investing in us, we actually are a very fast-growing startup, and right now we have 95 efficacy data for the COVID-19 vaccine data. Thank you very much.
Okay, thank you, Irene. Very informative talk. And I think due to the time, we may take one or two questions from the audience. I'll just call by the order. So I see the Yan Zhang raised the hand. Okay, Yan Zhang, now you are able to talk. You are on mute.
Or maybe you can collect his questions so you can repeat it to the audience.
Yeah, maybe maybe let's start from the question in the chat box. There is one question: any IP issue caused mRNA application? And cause mRNA application and mRNA application and LNP application. Are those
Okay.
patents? Yeah.
Oh, that's a good question. So basically, we build up a library. Instead of directly to modify the LNPs other companies are working on, we build a library to screen for new ones. So there's no IP issue. We already consulted many IP lawyers already, actually.
Yep, great.
And
great.
our own IPs have been well protected.
Okay, it's good to know. Okay, and I think consider the time, like we may stop here. And if you have further questions, please reach out to Irene, and her information is already shown here on the screen. And thanks again, Irene. And I will
Thank
say
you. Thank you very much for hosting.
Sure.
It's great honor to be here. Thank you.
Yeah. Thanks. Also, that's the end of all the roadshow we have today, and we would like to thank all of you all for your time and participation. And meanwhile, we also want to thank our general sponsors, Yifangda, Hengtian, VSM, Shanghai, Zhejiang, Boston Enterprise Park, and New England Capital, as well as our partners for broadcasting this conference. And last but not least, we also want to thank all the organizers, and thank you all for your contribution and make this come true. Okay, and yeah, so thanks again, and good night.
Bye-bye.
Bye.